Welcome everyone. Thank you very much. First, I would like to talk about bioinformatics. So why bioinformatics? This is our latest, newest master program, and we decided to launch this program because we see the need for scientists and professionals who can deal with this new kind and large amount of data that is continuously coming in from all kinds of biological experiments. So basically, there is a bottleneck in the world in terms of professionals who can analyze and meaningfully interpret all these data that are coming in from biological sources. What data types are these? Mostly genomic and transcriptomic data, but also we have data on epigenetic changes and proteomes. We are moving towards personalized and single cell data, so the resolution of these data is increasing, and there are projects in the world which aim to link anatomical information, structural information with molecular information. These will necessarily, inevitably need novel aspects and novel tools to analyze. And of course, protein structures, which is close to my heart. I'm a structural biologist, so that are needed to understand the molecular processes of life. So what can be the approaches? We can, of course, analyze single sequences if we know what we are investigating. We have a target gene that should be investigated. We can have large data sets like omics data, genomics, and transcriptomics data in which we need to apply sophisticated data mining tools. And we can also simulate scenarios, which brings us to the realm of systems biology. On the next slide, you can see an example of exploring human genetic variation. So there are large projects which aim at sequencing a large number of individuals, both healthy and both affected with some condition, and large data sets are accumulating which await basically the analysis by experts, and this cannot be analyzed by people who are not proficient in both the computer skills, both the data analysis aspects, and they should also know the biological background, otherwise these data cannot be interpreted. On the next slide, we can see some protein structures. This year's Nobel Prize in Chemistry was awarded for the breakthrough in protein structure prediction and protein structure design, which all utilize sophisticated computational pipelines. And although they do not solve all of the problems that are present, they still provide a huge leap in the field of structural biology. And in the next slide, you can see some examples of reaction networks and protein interaction networks that can be modeled using systems biology. This is a mathematical modeling technique with which complex systems like a full cell, for example, can be modeled. And these approaches are needed to understand how, for example, a mutation or the introduction of a drug can alter the functioning of a cell. What do we offer in this course? In this program, we offer some general courses like quantitative biology, chemical biology, programming, biostatistics. So the main point is to provide an advanced background both in molecular biology and cellular biology, both in programming. So you will get all the courses in these areas. And there are two specializations in this program. One is the so-called more classical specialization, biomolecular data interpretation, where molecular diagnostics is present and complemented with structural bioinformatics, proteomics. So the main point is that you can understand the molecular detail of a given mutational setting, for example, that you get in a clinical study. The other specialization is systems biology, which aims at the modeling. This has also some clinical relevance, as there are startup companies, even in Hungary, that aim to understand complex systems and their perturbation under several disease conditions. After graduation, of course, you can stay in research. That's always a possibility. We believe that we provide all the opportunities for that. We have experience with students who have a background in bioinformatics from other programs like Medical Biotechnology, from which I will talk about later. So our students are welcome at companies which need people with medical knowledge, biomolecular knowledge, and computer skills, and who can participate in molecular pipelines. The difference between this, I would like to stress this as a last thought. The difference between the bioinformatics master and, for example, the bioinformatics specialization in medical biotechnology is that the bioinformatics master is aimed at a level that is beyond the usual, so to say, user experience, so that even using bioinformatics tools professionally is a challenge. But we also would like to raise the stakes to provide the background in mathematics and programming for our students to be able to participate in the development of novel methodologies and novel analysis pipelines and novel tools in bioinformatics, so that they have the possibility. They don't have to do that, but they have the knowledge, they have the background to participate in such project. I think this is the end, and thank you for your attention.
Thank you very much, Professor Gaspari. So now the time has come to see your questions about the Bioinformatics Master's Program. Actually, in the Q&A channel, I cannot see any open questions right now. So let's see if someone comes up with a question. And if not, we will move on to the next presentation and see if you remember any questions that you may have during the next presentation.
I will be
the next presentation will also be delivered by Professor Gaspari, so he will definitely be here with us to answer your questions. Okay. We have a question. Is it possible to take courses online in bioinformatics? Well, the course is not online, it's in person. But maybe Professor Gaspari can tell us if there are any online courses as part of the master's program, or is everything here at the faculty with physical presence?
Yes, this program is for physical presence. We have other programs which are fully online, but they are not at the level of master program. We have a 1-year postgraduate certificate program if you are interested, but it doesn't provide any biology. It expects strong biology background, and it's only 2 semesters, so it's not a full master program. Industrial biotechnology, we should see your transcript, or record of transcripts, and see what you have learned. It's not impossible. We can discuss this when we see your exact courses that you have learned. We expect that students will have either a strong background in molecular biology or a strong background in computing. So if either of these are present, then it might well be possible to enter the program. Yes, ecological. How many companies actually give jobs on bioinformatics in Hungary? Several dozen, I guess. I don't know all the companies, but there are large pharmaceutical companies that supply people who are doing bioinformatics, and there are many small companies. There are a number of molecular diagnostics based companies, or companies doing molecular diagnostics who can offer jobs in bioinformatics. So there is a possibility to have a job there. Ecological issues are not really our cup of tea. So if it has some molecular aspect, so this bioinformatics course is about program is about the biomolecular aspect. So if there are no biomolecules, metabolites at least involved, then this is not what we can offer. Um, okay, medical background is possible. Yeah, yeah. You can contact us. So I guess I think we have contact address which we can interact. Uh, okay. Yes, if you have no experience in programming, you will have to take up some basic programming courses. That is possible when you come here. So it's we can do this because we will have this first semester that will offer some uh, courses that you can take up and fill the gaps that are required. Uh, Okay, I don't know what this head test score is.
I think this is the question that we will discuss in the second
Okay,
part.
okay, okay,
The
okay,
admissions procedure comes later on, so these questions will be answered in the
okay.
second part.
Yeah, yeah. If you need an HPC cluster, yes, we can have access to the Hungarian HPC cluster if needed for your research. Not for playing around with tasks, but yes, there is a. And we also plan to offer some basic training at regular courses, so we are working on that. But for research, definitely. Okay. I will do the comparison in the next with biomedical engineering. Uh, yes, I will answer that in the next presentation. Yes, regarding your previous studies, we should see your transcripts. For pharmacy, it's quite different from all over the world. Some pharmacy degrees include lots of molecular biology, others much less. So we have to see the exact subjects you have taken. Um, um, Yes, math credits are useful because you will learn some math, but it's not too strictly required. You can level up your knowledge. Okay. How many international graduates get jobs in Hungary? I cannot answer that question. Maybe Agi has some numbers.
We don't
Uh,
have numbers, but we do have a couple of graduates who are staying here in Hungary. But it's also quite common that they go on and look for jobs in other European countries. So
okay.
with these degrees, you can find jobs in other places in Europe as well. But some of them do stay here in Hungary if they get to like the country.
Okay. Uh, Yeah, Jedlik Laboratories is basically a research organization, so it's yes, you can join any research group that fits the profile of the program, of course, that is listed on the Jedlik Laboratories page. I think we should move on to the next
Yes, I
presentation.
agree because some questions are also coming up concerning medical biotechnology
Yeah,
and the link
yeah,
between the
yeah,
two
yeah.
programs. So I think it's
So
it's the time that we start the presentation about the medical biotechnology, and then we can
we
answer
will
the remaining questions
come
after
come back
this.
to the questions. Yeah. Um, so medical biotechnology uh, is a bit older master program. Why medical biotechnology? Because in not only the informatics part of the biomedical sciences is changing fast, but also all other kinds of molecular level aspects. So yes, we have data, but we have also emerging technologies to manipulate macromolecules and complexes and to use them for diagnosis and therapy and to modify cellular systems. So our toolkit is increasing to do this to use more and more advanced diagnostic techniques and more advanced therapy techniques. They are not. They are moving hand in hand, and the so-called the molecular and cellular manipulation tools and aspects are rapidly expanding. So for molecular biology, you need to know if you want to be a proficient in the emerging medical technologies in biotechnology. So you have to know how to manipulate biomolecules, how to isolate them, how to investigate them, how to build something that you need. This is already bionanotechnology. With bionanotechnology, we can build molecular devices and molecular um, um, assemblies that can be used for drug delivery, for diagnosis, and for targeted manipulation of cellular systems. And of course, yes, part of the story is computer skills. The major difference between the bioinformatics offered here in medical biotechnology and the bioinformatics master is that here you will only get a somewhat limited set of bioinformatics, which is required for a daily work for someone who is basically associated more with the laboratory. So some for people who are doing lab work also need some data analysis, also need to understand some aspects of data analysis. This is provided, or if you do some routine, more or less routine bioinformatics analysis, but don't want to go into the deep details of the algorithms and don't want to be a developer of bioinformatics algorithms, then this applied bioinformatics specialization might be for you. So it depends on how exactly you position yourself between lab work or actual medical work with patients and the analysis pipeline. So the bioinformatics is for people who specialize in the analysis and want to even be developers and want to understand all the deep aspects. This is for more or like a user perspective. You will know what tool to use for what, and but will not be able to implement larger modifications, for example, in an analysis pipeline. Not necessarily be able, because this course, this program will not provide such deep programming skills for you. Okay, on the next slide, we can see how our understanding, for example, of the human genome is changing. So these two gentlemen are the medicine laureates from this year who got the Nobel Prize for the discovery of microRNA, which is an RNA-based regulatory mechanism. And there are estimates from last year that besides the approximately 20,000 protein coding genes in the human genome, we might have even more so-called long non-coding RNA molecules, which also participate in the regulation of genome. So it's an open question how many long non-coding RNA genes humans have. We just got started to think that we already know everything about the human genome. Then an aspect like this comes in, and everything again becomes open. So it's a very, very fascinating novel aspect, this RNA-based regulation. On the next slide, you can see the Nobel laureates from 2020, Jennifer Doudna and Emmanuel Charpentier, who discovered CRISPR. You surely heard about CRISPR. CRISPR makes it possible to manipulate the genome of cells and organisms with high precision. So in a genome that is so huge as the human genome, you can modify a single position with this tool because it provides you so precise targeting and is relatively easy to do in the lab. So gene therapy, the current generation of gene therapy tools is based already on CRISPR, and this is already not the future but already the present, using such kinds of therapies. And again, on the next slide, you can again see two Nobel laureates from last year, Katalin Karikó, who is Hungarian, and his colleague David Weissman, who contributed to the development of messenger RNA-based vaccines, which is basically a field of bio-nanotechnology. So they had to modify the messenger RNA properly and package this messenger RNA into a particle that can deliver this messenger RNA into the cells and use the necessary answer. And what the Nobel Prize was about is that they had to balance some effects of this messenger RNA. It had to produce the protein against which the immune system will react effectively, but also should be recognized to some extent as a foreign material to make the immune system alarm. So it's a delicate balance. So on the next slide, I think I already summarize what we have here. As courses, a highly important aspect of the medical biotechnology program is that it's in close collaboration with Semmelweis University, which is a medical university. So many courses I taught by teachers from Semmelweis University, and you can choose many research topics that are pursued there in a clinical setting close to patients. So that's very important for this program. We teach advanced molecular biology, biophysics, recombinant DNA techniques, tumor biology, stem cell biology, signal transduction. So all kinds of topics of advanced molecular and cellular biology. We have two specializations: molecular biotechnologies for those who would like to work in the wet lab with cells or molecules, and applied bioinformatics is for those who would like to know more about the data analysis and processing. Again, this is more like a user perspective and not the deep developer perspective with understanding all the subtle details that is offered in the full bioinformatics master. So it's for more biology oriented people who are closer to the lab or clinics. And our uh, previous graduates, many of our previous graduates still work in bioinformatics related tasks at companies who perform analysis of medical data or personalized medicine or molecular diagnostics. So this is a possible career path, but also many students work in laboratories. It's a very high fraction of our significant fraction of our students pursues a PhD after graduation. So this might be one of the programs in which the highest percentage of students pursues a PhD. So with this, I would like to thank, and if there are any specific questions, I will try and answer them.
Thank you very much again, Professor Gaspari. And let's see if there are any questions related to medical biotechnology.
Okay, so I'm starting with the last one, focus area of research in bioinformatics. So for example, we are doing structural bioinformatics. That means protein structure analysis and comparison, uh, prediction of the occurrence of specific structural elements in genomes. We have a systems biology research group in which we work with, for example, the prediction of protein complexes given a set of proteins and try to explain things that are there that happen. But also this group also works with the cell cycle regulation, and we have research groups which are using, for example, language models to analyze genomic data to recognize specific patterns in next generation sequencing data. So we are really, I think we cover a very large palette, and besides our own research groups, we have collaborations with research group at Semmelweis University who work with problems more directly related to molecular diagnostics and things like that. Okay, so for any specific questions about previous studies, I can only repeat myself that we will have to see your transcripts. Study R and different programming languages. Yes, R and Python are part of the courses. Yes. What is the research that is conducted right now in your in medical biotechnology? It's very hard to answer because there are many research groups also in Semmelweis University. So there are research going on tumor cell lines, how they respond to treatments. We, in my lab, we are working on proteins in synaptic transmission. It's a protein level study. Other many projects involve data analysis in diagnostic settings or some biophysical studies of proteins. So there are really lots of research going on DNA analysis, molecular data analysis, or next generation sequencing analysis. So or transcriptomic analysis. So there are really uh, wide choice of microRNA-based regulation or extracellular vesicles are part of the topics that are pursued right now. Yes, so again, the background we will have to see the exact transcripts to say yes or no. But in general, if you have a strong background in molecular biology or have a strong background in programming, it might be possible to take up courses that will make you competent in the course.
I can see one more question.
Yeah.
And maybe that if there are no more questions, then that could be the last one. Focus area of research in bioinformatics. It's going back to the first presentation.
Yes, that's I try to answer because we have multiple research group at our university, and again we have cooperations with Semmelweis University. So we are working with next generation sequencing data. We are working with data that are used in systems biology modeling, protein abundance data, genetic regulatory networks, and we are working also with protein structures. So all kinds of data are used in our research. So monoclonal antibodies, I don't know about any program that would focus on the production of monoclonal antibodies, but many research uses monoclonal antibodies as a tool, also here at our university and at Semmelweis University. Yes, I think I answered all the entry questions that I was competent in. You cannot really do thesis work in other countries. I would say no to that because you have to be here with your supervisor to do that. So unless the only possibility is that you can have a specific scholarship like an Erasmus and have a joint supervision. But it's very hard. I won't recommend doing entirely research abroad because then we have no control about it. So you have to be here physically to do that.
Okay, thank you very much. As I can see, there are no more questions related to the professional content. All the questions that I can see can be answered by us in the second half of the online open day. So thank you very much, Professor Gaspar, for both of the presentations and for answering the questions.